In this work by Álvaro Herrero Navarro and colleagues of my laboratory, we have found for the first time a molecular signature that is co-expressed and shared by cells that are born in the same brain region. We have found that neurons in the brain and astrocytes, which are a type of glial cells, co-express molecular signatures that are well recognized as neuronal markers for a long time. So these signatures expressed by the astrocytes can be used for brain repair. So what we have found is that these astrocytes can be used to introduce a master transcription factor with reprogramming properties, as for example neurogenin 2. And because the different astrocytes of the brain express these molecular signatures that we have found, we can reprogram them both in vitro and in vivo into specific subtypes of neurons. As I said, this has important brain repair meaning because we can now use these astrocytes and this imprinting that they have to reprogram a specific subset of neurons, sensory neurons in this case, which have been lost in sensory deprived animal models, for example. So we know that in congenitally blind individuals, and also has been replicated in animal models, so when one of the input from the sensory periphery is lost, many neurons and circuits are being also lost during life. So we can use these molecular signatures that we have found that are hidden in these astrocytes of the brain for reprogramming sensory circuits as those, for example, that are necessary to be recovered in sensory deprived individual, as for example, congenitally blind or congenitally deaf. And this is important for recovering in the future the senses that has been lost, as for example, vision.